Dr. Orville Baldos. He is an associate researcher in sustainable ornamental production at the Department of Tropical Plant and Soil Sciences, University of Hawaii at Manoa. His current research focuses on developing selections of native Hawaiian plants for landscape and ornamental use. He is also interested in screening and selection of non-invasive ornamental species and developing sustainable ornamental plant production and landscape management practices. He received his BS in agriculture, specializing in landscape horticulture from the University of the Philippines, Los Banos, and both MS and PhD degrees in tropical plant and soil sciences at UH-Manoa. Dr. Baldos.
All right. Thanks for inviting me to this seminar. So today I'm going to give you some updates from the past year on our research on native Hawaiian ornamentals. Just a little bit of an outline of what I'm going to discuss during this presentation. So I'll be covering germplasm release for this year, some updates on where I distributed these materials, and some research updates on Peperomia sanduchensis, micropropagation, indoor light tolerance and of new selections, trying to propagate variegated naupaka, also trying to identify ways to break seed dormancy of Carex wahuensis, and exploratory projects such as looking at a grass, so this is alpine hair grass called Descampsia nubigena as a potted plant, and doing some germplasm collections of variegated natives, and then a little bit about weed steaming because we have an upcoming project on steaming. Okay, so first off, we released a new selection of Jack Montia or Paohiaka, and we named it after Leeward Community College. This publication came out in Hort Science, so this is our official germplasm release publication. I also did a magazine piece on Hawaii landscape to notify this germplasm release to the landscape industry. So Jack Montia, Leeward Community College white. The origin is from a plant that was growing at Leeward Community College. We got it as a seedling from Lion Arboretum, and when we saw that this plant has nice ornamental characteristics, we kind of tried to go back and see what the history is, and the furthest that we could go was Leeward Community College. So this plant has 10.5 millimeters in diameter flower, white flowers, green leaves, no hairs at all, and then the leaves are oval. The stem is purple and has medium pubescence. And right now we're distributing cuttings or plants to interested groups. Here's a picture of LCC in a hanging basket plant. So this was done by one of my master students back in 2018. Next would be germplasm distribution update. So since I've done research on native Hawaiian plants, we've released 3 selections of native plants, so 2 selections of Jack Montia and 1 selection of Peperomia. So the first selection of Jack Montia that we released is Puhala Bay. This selection is a Maui accession. So we distributed this material this past year. We have distributed 30 potted plants and 81 cuttings. Recipients were 2 botanical gardens on Oahu, 4 nurseries on Oahu, Hawaii Island, and Kauai, and 2 hotels and resorts in Hawaii and Oahu, and then 3 individuals who were hobbyists. Okay. Next is Leeward Community College white. So we distributed 13 potted plants and 81 cuttings. to 1 botanical garden on Oahu, 4 nurseries on Oahu, Hawaii, and Kauai, and then 2 hotels and resorts on Oahu and Hawaii Island, and then 1 hobbyist. And then the peperomia that we released last year, Peperomia sanvichensis pallikea. This was distributed this past year, so we distributed 4 potted plants and 27 cuttings to 1 botanical garden on Oahu, 3 nurseries located on Oahu, Hawaii, and Kauai, and then 1 hobbyist. So we're trying to figure out, so we've figured out how to propagate pallikea using leaf cuttings and stem cuttings. Now we're moving on to trying to mass propagate it through tissue culture. So one of the projects that one master student did was trying to figure out a sterilization technique for explants that had like a single node with leaf and a single node without the leaf. So as you can see here in this slide, we had sterilization using Physan on top. So the first 2 rows is Physan. We didn't get good results in terms of less contamination. So in fact, we had more contamination with just Physan only. And then at the bottom, Physan and Clorox. So sterilizing it in Physan for a couple of minutes and then Clorox next yielded more sterile explants. So we had greater success with Physan and Clorox. So based on this student study, we recommend, so if you're going to do tissue culture work with Peperomia sanvichensis, I would suggest growing it indoors under light conditions, so just to reduce the microbial load. And then once you've grown it indoors, get the explants and soak it in sucrose for 5 hours. And then after 5 hours, wash it off, do a Physan soak for 1 hour, and then wash off, and then Clorox, and then rinse, and then that should be ready for putting in culture. So we tested explants that contained a leaf and then without the leaf, and we didn't really see any difference in terms of the number of shoots. We only saw like subjectively like vigorously growing shoots with explants with a leaf, but it didn't really had any difference in the number of shoots or roots that are produced with the explant. So currently we're preparing this one in publication, so hopefully we'll be able to publish this in a journal. And right now we have been able to place Peperomia pallikea in culture. So I think right now we have about maybe like 9 magenta boxes. That's like this much. So we're going to do subculturing soon. So we're trying to increase the availability of microcuttings so that microcuttings could be another way of propagating this peperomia. So we're currently looking for interested growers who might want or who have the capability of doing in vitro propagation, and we can like hand over maybe 1 magenta box, and you guys can start that one as your stock plants for in vitro propagation. So yeah. Okay, next area of update is a new selection of peperomia for testing. So this is a same species, Peperomia sanvichensis, but this one is a bigger leaf accession. So this one was collected on Oahu, and we got the material from Lion Arboretum. So it has bigger leaves compared to pallikea and has more pronounced leaf veination. So we were able to successfully put this in sterile culture in March 2018, and we've been increasing the subcultures ever since. And this was actually the study species that the study selection that we used for trying to figure out acclimatization studies. So trying to produce microcuttings, and then what do you need to do with those microcuttings outdoors to make it successful. So this is the other work of our master's student. So what she did was she collected microcuttings, and then we tested different types of growing media. We also treated the microcuttings with or without rooting hormone. And basically what we found is that 3,000 parts per million of indole-3 butyric acid can improve rooting, but it's optional. It doesn't really have an advantage in speeding up growth of the plant. We found when we tested different types of growing media, we found that planting apical microcuttings in sunshine mix number 4 had the best results. We suggest to grow the microcuttings. So once you've planted those microcuttings, grow them under indoor conditions in LED lighting for about 4 weeks to allow it to root. And then after 4 weeks, you can move it in a greenhouse and grow for about 7 months until you reach desired size. So the picture at the bottom is the 4-month-old plants from microcuttings. So right now we're testing indoor light tolerance of this particular selection. So we just started the trial last month, and we're testing 3 different light levels. So the high light level, which is 5,000 lux, office light conditions, which is 750 lux, and low light conditions at 350 lux. We'll be looking at qualitative and also quantitative characteristics like height and diameter. And this will end in February 2023. So I have a student that's working on it for the next 3 months. Okay, another update, which is looking at propagation techniques for variegated naupaka. So in recent years, there have been a number of variegated naupakas that have been seen on different islands. So there's one that I saw in a botanical garden on Oahu, which the origins are from Molokai. And then I got an unnamed variegated accession from Kauai Nursery and Landscape. And then Hawaii Island, so this island also has a variegated naupaka, and it was named Fukupaka. It was published in Hawaii Landscape magazine, I think, back in 2019. And then one nursery on Oahu has identified or selected a variegated naupaka for Oahu, but this isn't for sale yet. So, so it's not rare. So we can have variegated naupaka on different islands, but right now, so they're available, but it's not being used yet on a large scale. So the only place that I've seen it used on a large scale was at a resort on the Kona side, because the Fukupaka was actually identified on that resort, and they just propagated it, and then they used it in their landscapes. So in order for the variegated naupaka to be available, we wanted to look at propagation techniques. We tested the use of IBA on this particular selection. So this was collected from Kauai via Kauai Nursery and Landscape. So variegated naupaka, it's a lot more. So since it's variegated, it's a little bit compromised in terms of photosynthesis. So it's a slow-growing plant, and you can actually maintain like dwarf plants as a potted plant. So it could be used potentially as a container and maybe as a low maintenance landscape plant. So as I've said, material is there, but it's limited in terms of numbers, so it's not yet widely available. So we wanted to test how the effect of rooting hormone if it can improve like rooting characteristics of cuttings. So we tested the Kauai selection. So we got 8-inch apical stem cuttings, and we removed the lower halves, and then we tested, we treated it with different amounts of IDA, indole butyric acid, and collected rooting data about a month after we applied the rooting hormone. And what we found is that rooting hormone, regardless of concentration, so the lowest concentration that you can apply rooting hormone and get good rooting is about 2,500 parts per million. So it increased total root surface area and root length. But when we compared the concentration, so 2,500 versus the concentrations, increasing concentrations, we didn't see any significant difference. So you can just use the lowest concentration and get the same result for rooting. So the minimum is 2,500 ppm. And our plans for this year is to repeat the experiment and prepare this for another publication. This past July or August, I presented this result in the ASHS conference, so it's available as a poster, and I'll post this soon on my website so people can access. And next project that another master student in our lab is doing is trying to figure out a way to hasten germination of Carex wahuensis. So you all know maybe Carex wahuensis is an endemic ground cover. It's an important species for revegetation, also for landscaping. And it's typically propagated from seeds, but it takes about 6 to 10 months to get germination. You can divide Carex, but it's very slow and difficult. Like you can't divide single plantlets, so we've done that, and the plants die on us. So bigger clumps is more successful, but single clumps isn't. And when was this? Probably around 2020. Huiku Mauli'ola, which is a native nursery on Oahu, they released a variegated selection, and they're selling it really high price, like $30 per plant. So we wanted to find out ways to propagate the variegated plant by seeds. So knowing what seed treatments would work is one step or not, because we don't know whether the seeds that we get here will have variegated plants. So knowing that seed treatment is one step towards increasing the variegated form. This is just a picture of the variegated Oahu sedge, because Huiku Mauli'ola got, I guess, the contract to landscape Turtle Bay Resort. So I was able to find, when I was there, I was able to see that, oh, they're using it in the landscape now. So to figure out a way to increase germination or shorten the germination time of Carex wahuensis, we looked at the literature, and we found that temperate species of Carex benefited from long-term cold exposure, like months in cold. But we don't have it here, so probably a short-term cold exposure would be what makes sense with Carex. So we tested short cold exposure treatments, and we found that by putting the seeds in a refrigerator, so 46.4 degrees Fahrenheit, and as high as 58.1 degrees Fahrenheit. So when we had that cold dip actually last December, I don't know if you guys remember that, like really high rain event and then followed by a cold surge. We got stimulation and germination with that cold snap. In terms of the most effective period of time in terms of exposing the seed to cold temperature. So if you sow the seeds under ambient conditions and then after 3 or 4 months you expose it to cold temperature for about 3 days, about a week after you get some germination going on. So our next steps for this project is trying to identify what the minimum temperature is for germination. So what's the upper end of the low temperature. This study is actually part of one of my master's students' thesis, and she will be defending this one this fall, and will be presenting this work and other parts of the work on Carex. So this result was also presented at the ASHS conference in Chicago last July, and then I'll be uploading this one, this poster soon on my website. Some exploratory projects. So I'm also interested in looking at native Hawaiian grasses in landscaping, because I don't see much grasses being used, like Hawaiian grasses being used in landscapes. And one species that kind of caught my eye in terms of potential as an ornamental is the Scampsia nubigena, or alpine hairgrass. So alpine hairgrass is actually endemic to the high mountains, so Mauna Loa, Mauna Kea, and also Haleakala. We got seedlings from Lion Arboretum, and the seeds trace back to Hawaii Island. This was back in 2020, and as you can see here, it has a very short stature, very fine, almost like cylindrical leaves, this tall. Kind of looks like mondo grass, but really more fine in terms of texture. So I can see it used in a potted situation and also as a landscape plant. So in order for this selection or this species to be useful in the landscape, we need to know propagation techniques. If it's easy to propagate, then it might be useful. So we found that this species can be propagated from division fairly easily. So we're going to do more replicated studies on propagation, and then hopefully have a landscape and potted plant trial in the next couple of years. So picture on the left is the plant in its natural habitat. So this is on Haleakala. And then the picture on the right is our potted plants growing at the Magoon Research Station in Manoa. We haven't observed any flowering in the past 2 years, so I think it's more of a cold, it might be, the flowering is stimulated by cold. So here's a picture of the Scampsia being divided into like single plantlets versus clumps. And about 2.5 months after division, we have good regrowth. So this is in like 2-inch pots. So with very minimal mortality. So has potential to be propagated easily. Another plant that we're looking at is this nice, so there's the white variegated Scaevola tacada, and then there's this like lime green variegated Scaevola or naupaka. This was actually isolated from the variegated naupaka that we got from Kauai. And interestingly, like in Fukupaka, I was able to go to the place that they collected the Fukuoka, they actually were able to isolate the same type of variegation in that variegated Fukuoka. So it's not, I guess this type of variegation is not like rare. So some updates, we're currently increasing this plant, and then seeing whether the variegation is stable enough. And we're seeing like some stability on it. Like there's like maybe a few branches that are reverting back to green. A student will be looking at IBA on propagation of this particular selection later this year. And I've just propagated a couple of cuttings, and we'll be comparing this one with the variegated form, the white variegated form, and then the wild type, which is the non-variegated form, and then see whether how it compares in a landscape setting or as a potted setting, in a potted setting. Then we were able to get the variegated Carex from Huikuma oleola, so we have a collection of in a pot growing in our benches. So right now we have collected seeds of this variegated accession, and I'm basically testing out the cold treatment protocol and see how it fares, and if we get germination, and then at what percentage or like the number of plants that are variegated, if it's passed on to the seeds. So the goal is basically to develop propagation protocols for nurseries. If the seed propagation doesn't work, then maybe we'll look at tissue culture work. So I need a student for that and also funding. So the goal is to propagate and distribute the plants to the nurseries to make it more available. So this past summer, I was able to go to the Big Island and was able to collect Fukuoka, which is a variegated Naupaka on the Big Island. So this was a selection from the Kona coast, and we need to check whether the plant has viruses, and because I can see that there's some leaf crinkling in the plants that we collected. So I just wanted to make sure that the viruses are, well, the plant is clean before we propagate and distribute, yeah. And next steps would be to identify propagation techniques for growers and maybe compare, because there's some differences that I see with the Kauai selection and then the Fukuoka in terms of characteristics. So maybe do some comparative characterization of both. I was also able to obtain a variegated vitex, beach vitex, Pohinahina, on the same property. The person, my contact person there, was gracious enough to give me some cuttings. He just warned me that the variegation might be unstable. So what we'll do is we'll grow it out and then see how stable it is. And if it's stable enough, then we'll check for viruses and then do landscape trials and characterization, and then eventually propagate and then release this to the landscape industry for use. This is what the plants look right now in the nursery, since we collected it in June. So as you can see, the variegated beach vitex is a little bit more slow growing. So it's, I guess its invasiveness is sort of like tamed a little bit. Okay, a little bit of a different route with my research update. So I was also able to get funding to purchase a weed steaming equipment for Lion Arboretum, and I'm working with them to find a way to do a non-chemical weed control. So trying to find or trying to identify which species can be controlled by steaming, what type of duration, and then what growth stage. Is it effective? I'm also trying to pitch this out to the other researchers here because this is a portable machine. It's just expensive, but once you have it, then you can do a lot of research, because it's a hot shower. So it's basically a hot water shower. It's like boiling water. It's and then you attach the machine to. So that's a portable machine. So it's a diesel-powered machine. Well, actually, the engine runs on a gas, and then the burner runs on diesel. Then you just attach a hose to the machine, and then it basically is like a hot water pressure washer. So it might have applications in terms of like nematode control, or like portable hot water shower for controlling mealybugs. And this last photo here is just showing how, I guess, effective the hot water treatment is. So this is just one day after like a couple of hours after application. So we kind of killed the turf grass here. And this is about 2 weeks after. So it's effective in killing some turf grass species. All right, I'd like to thank the grad students that I've worked with, my student help, different collaborators, and my funding sources. And if you have any questions, you can email me, you can visit my website, or look at my Instagram page for any updates. Thank you.
Any questions for Dr. Baldos?
Yes. So the question is, can you control the temperature of the pressure washer? Yes. I guess I have to check what the lowest limit is on that pressure washer. But definitely it can go like boiling, like 150 Celsius, something like that. So.
Have you ever run freeze-dried trials? Also with the alpaca?
So have I ever run base life trials on the variegated alpaca? I've done just one cutting with like mature ones, and it kind of lasted for several days. But I guess if it's like succulent growth, like really soft, it doesn't hold up. But if it's more like woody growth, then it kind of holds up the foliage.
Have you ever ran cut foliage trials on that?
We haven't run, so question is the variegated carex, have we ever done any cut foliage trials? We haven't, but on the non-variegated ones, it lasts a long time, but the problem with the carex is that it can cut you. So if there's a way to remove, I guess, I don't know, through maybe breeding or maybe increasing ploidy levels, we can make it like not sharp, softer. That might be useful.
I was just curious, how much does that fresh or hot water machine or setup
Hot water.
cost?
So the question is, how much is the hot water machine? It's $20,000.
All right. Thank you very much, Dr.
Thank you.
Baldos.